Greetings, dear students. This is part 3, Nervous System Physiology. I'm Assistant Professor Ghaithali Jasim. Today we will talk about the transmembrane potential. There are two types of transmembrane potential. First is called the graded potentials, and the second is the action potential. So first let's have some information about the graded potentials, or called local potential. The definition is the changes in the transmembrane potential that cannot spread far from the area surrounding the site of stimulus. In this case, the membrane is exposed, for example, to a chemical that opens, for example, chemically regulated sodium channels. So sodium enter the cell, and additional positive charge shifts the transmembrane potential towards 0 millivolts, towards the positivity. So any shifting of the transmembrane potential away from the resting level, if you remember it's minus 70 millivolts, towards 0 millivolts or above, it's called depolarization. So we have a new term now. It's depolarization. It's shifting of the transmembrane potential away from the resting level towards 0. Okay, this nice image of a drop that caused some waves. I want you to put this in your mind. This you can remember the definition of the graded potentials. Let us continue. The movement of sodium ions across the cell membrane at one location causes the depolarization of the surrounding membrane. So sodium ions entering the cell spreads along the intersurface, attracted by the excess of the negative charge inside the cell. The ion movement in the inner and outer surface membrane is called local current. The change in the transmembrane potential and the area affected by the local current are directly related to the number of sodium channels open by the chemical stimulus. So the more channels open, there will be increase in sodium spread, and then finally increase depolarization. On the other hand, opening of potassium ion channels will have the opposite effect. So potassium will flow out of the cell, and the inner surface of the cell will lose positive ions and produce a state of hyperpolarization, which is a shifting of the resting potential towards, for example, minus 80 or minus 90, towards the more negative. And also a local current will distribute the effect to adjacent cell membrane. Now I have a nice image of two drops that cause two waves that could interact between one another. This is also a clever way to understand graded potentials. So what are the characteristics of graded potentials? Let's put in mind those drops that I talked about. The transmembrane potential is most affected at the site of stimulus, and the effect decreases by distance. The effect spread passively due to a local current. The graded potential changes may involve depolarization or hyperpolarization, depending on the membrane channels involved, sodium or potassium. The stronger the stimulus, the greater the change in the transmembrane potential, and the larger the area affected. The second type of potential is the action potentials. Action potentials, the definition, they are propagated changes in the transmembrane potential that once initiated spread across the entire excitable membrane. An action potential generally begins at the initial segment of the axon. The action potential is then propagated along the length of the axon, reaching the synaptic knobs. So this is an illustration for the axon, and the blue color resembles the generation of the action potential and how the action potential spreads from segment to segment in the or across the length of the axon. So the stimulus that initiates an action potential is a depolarization large enough to open voltage regulated sodium channels, and the opening occurred at the transmembrane potential called the threshold, which is -60 to -55. All stimuli that bring the membrane to threshold will generate an action potential. So the properties of an action potential does not depend on the strength of depolarizing stimulus, the graded depolarization, as long as the stimulus exceeds threshold. This is called the all or none principle, because a stimuli either trigger a typical action potential or does not at all. So this figure shows us the voltage and time and how the action potential is generated from simple depolarizations, or here it's called stimuli. So whenever these stimuli reach threshold, the action potential will start. Action potential generation. First step, depolarization to threshold. Before an action potential begins, an area of excitable membrane must be depolarized to threshold by a local current. Second step, activation of sodium channels and rapid depolarization. At threshold, the activation gates open and the cell becomes much more permeable to sodium by the effect of the electrochemical gradient. Sodium rush into the cell or into the cytoplasm, and rapid depolarization occurs. So from this figure, we can see in the blue circle, this is the depolarization to threshold. From -70, a stimulus or a depolarization or a graded depolarization brings the transmembrane potential to threshold. Then there will be activation of voltage regulated sodium channels. The activated gates will open, and then we will have the depolarization step of the action potential. Sodium channels inactivation and potassium channel activation. This step at transmembrane potential of positive 30 millivolts, the inactivated gates of voltage regulated sodium channels begin closing. This step is called sodium channel inactivation. At the same time, potassium channels are opened. The last step of the action potential generation is return to the normal permeability. Voltage-regulated sodium channels remain inactivated until the membrane repolarize to threshold, which is about minus 60 millivolts. Then they remain closed but capable of opening. Voltage-regulated potassium channels begin closing as membrane reaches normal resting potential, which is about minus 70. So action potential starts at threshold. There will be activation of sodium channels, voltage regulated sodium channels, and rapid depolarization. Then there will be sodium channel inactivation and activation of voltage regulated potassium channel. There will be repolarization and return to the normal permeability. How can the action potential be propagated along the length of the axon? Action potential will begin at the initial segment, which is the first segment of the axon following the cell body, the soma. The membrane is a series of adjacent segments. The axon hillock cannot respond to an action potential. The axon hillock is the part between the cell body and the initial segment of the axon. At peak of action potential, the transmembrane potential will become positive and a local current develops. Sodium ions move in the cytoplasm and in extracellular fluid, and the local current spreads in all directions, depolarizing adjacent portions of the cell membrane. The process continues in a chain reaction or as tiny steps along the parts of the axon. So we can see from this figure how the action potential in deep red, how it's generated, and sodium will spread along the length of the axon. So each time a local current develops, the action potential moves forward because the previous segment of the axon is still in the absolute refractory period. So action potential always proceeds away from the site of generation and cannot reverse directions. We can see that in the area following the blue action potential part on the axon below. The area is colored green. This part where sodium channels are inactive, they remain inactive, and the activation is only for the potassium channels. So the inactive voltage regulated sodium channels, or we can say the channels are closed and incapable of opening. This is the refractory period. So to review the action potential propagation, we can see in this illustration, in the box number 1, sodium channel locally open in response to stimulus, generating an action potential in this area. Number 2, some depolarizing current passively flow down the axon from the deep red towards the more lighter red color. To the image below, number 3, local depolarization causes neighboring sodium channels, the voltage regulated here, to open and generates an action potential in the adjacent segment. We can see the movement of the deep red color to the adjacent segment. And in number 4, the upstream sodium channels inactivate while potassium channels open, the voltage regulated potassium channels. The membrane potential repolarize and the axon is in the refractory period. That means sodium channels are inactivated, closed, and incapable of opening. What about the myelinated axon? In the myelinated axon, the lipid content of the myelin blocks the flow of the ions across the membrane. So ions cross the cell membrane only at the nodes that respond to depolarization. The action potential in the myelinated axon is propagated 5 to 7 times more rapidly than in the non-myelinated axon. We can see in this illustration how the cell membrane is exposed only at the nodes. It's called the nodes of Ravier. In the red color we can find action potential generation, and in the lower image we can see jumping of the action potential to the next exposed cell membrane in the nodes. So a series of Schwann cells sheet block ion movement. So action potential must jump from node to node. This is a nice picture for a small boy trying to throw a rock. So we can see that wherever the rock touches the surface of the water, there is this wave again, which I called it the depolarizations or the initiation of action potentials along the myelinated axon. Okay, another subject here, the synaptic transmission. In the nervous system, messages move from one location to another in a form of action potentials along the axon. These electrical events are also called nerve impulses. A message must be transferred in some way to another cell. At a synapse involving 2 neurons, the impulse passes from the presynaptic neuron to the postsynaptic neuron. A synapse may involve other type of postsynaptic cells, for example, a muscle where there is a neuromuscular junction. A synapse may be electrical with direct physical contact between the cells, or chemical involving a neurotransmitter. We can see in the illustration below, when a nerve impulse or action potential arrives at the presynaptic membrane, this neuron will release a neurotransmitter which will connect to its specific receptor at the postsynaptic membrane. This receptor is located on a membrane channel. The connection of the neurotransmitter to its specific receptor will cause opening of the channel on the postsynaptic membrane, allowing the entry, for example, of sodium ions, and there will be depolarization for the postsynaptic membrane, and the signal will be converted from one neuron to the other. So a chemical synapse involves release of a chemical or a neurotransmitter from the presynaptic membrane to the postsynaptic membrane, where this neurotransmitter engages with a specific receptor, causing activation of a specific type of channel. So, what are the events that occur at a cholinergic synapse following the arrival of an action potential at the synaptic knob? This may be summarized by 4 steps. Step number 1, arrival of action potential, depolarized the synaptic knob and the presynaptic membrane. Step number 2, calcium ions enter the cytoplasm of the synaptic knob. Then a chemical or neurotransmitter, here acetylcholine, release occur by exocytosis of the neurotransmitter vesicles. Step number 3, the acetylcholine, the neurotransmitter, binds to receptor on the postsynaptic membrane. The chemically regulated, for example, sodium channels, are activated, and graded depolarization will occur to the postsynaptic membrane. The ACH release stops because calcium ions are removed from the cytoplasm of the presynaptic membrane. So, step number 5, depolarization ends as the neurotransmitter acetylcholine is broken down into acetate and choline by a specific enzyme called acetylcholine esterase. The synaptic knob reabsorbs choline from the synaptic cleft and resynthesize the acetylcholine, the neurotransmitter. A new subject here, the cellular information processing. At each synapse, an action potential arriving at the synaptic knob triggers a chemical or electrical event that may affect another cell. Some of the neurotransmitter arriving at the postsynaptic cell may be excitatory, while others from different presynaptic neurons may be inhibitory. We can see from this illustration that presynaptic impulses from the green color can be excitatory, the blue one, the black one, while from a different presynaptic neuron it could be inhibitory. The result may be depolarization or hyperpolarization, changes to the transmembrane potential at the initial segment. So the transmembrane potential at the initial segment is an integration of all excitatory and inhibitory stimuli. The excitatory and inhibitory stimuli are integrated through interaction between the postsynaptic potentials. The interactions are the simplest level of information processing. We can see here there is an integration of depolarizations and hyperpolarizations at the same neuron. So this integration of positive and negative, or depolarization and hyperpolarizations, will be the simplest level of information processing at a level of a single neuron. So the postsynaptic potential, they are graded potentials that develop in the postsynaptic membrane in response to a neurotransmitter. They could be divided into EPSP, which are excitatory postsynaptic potential. The definition is a graded depolarization caused by an arrival of a neurotransmitter at the postsynaptic membrane. The excitatory postsynaptic potential is caused by opening of chemically regulated sodium ion channels. The excitatory postsynaptic potential affects the area immediately surrounding the synapse. An example for the neurotransmitter that cause opening of chemically regulated sodium channels is acetylcholine. While the inhibitory postsynaptic potentials, the IPSP, is defined as the transit hyperpolarization of the postsynaptic membrane. The inhibitory postsynaptic potential may result from the opening of chemically regulated potassium ion channels, while hyperpolarization continue. Put in mind that the neuron is inhibited. In the transmembrane potential, where the resting is at, for example, minus 85 millivolts, because of the presence of the inhibitory postsynaptic potentials, the stimulus will depolarize the transmembrane potential or the membrane only to, for example, minus 75, which is still below threshold. That's why we said that wherever there is a hyperpolarization, the neuron is inhibited. Here we can find an image of an electron microscope showing the synaptic terminals, hundreds of synaptic terminals of presynaptic neurons connected to a cell body of a postsynaptic neuron, and to the left we can see the illustration of the neuron and different excitatory synapses and inhibitory synapses. We can see that in red and green colors. All these synapses will integrate to a final transmembrane potential, which if was exceeding threshold, it will cause an action potential to this neuron, and if it's not, the action potential or the signal will not be transmitted. Still on the subject of the postsynaptic potentials, let's understand the term summation. Summation is the mechanism responsible for the integration of excitatory postsynaptic potentials, inhibitory postsynaptic potentials, or some combination of the two in the postsynaptic neuron. There are two forms of summation: spatial summation and temporal summation. The spatial summation occurs when simultaneous or at the same time stimuli at different locations have a cumulative effect on the transmembrane potential. This involves multiple synapses. More than one synapse is activated at the same time. All of these will pour sodium ions across the postsynaptic membrane. As an effect on the initial segment is a cumulative. Degree of depolarization depend on the number of synapses active. So the more activated, the more depolarization that happen and could bring us to threshold, and action potential will generate. So we can see from this illustration, in A it's a normal action potential, with a single synapse between pre and postsynaptic neurons. We can find in B there are multiple synapses and action potential are arriving all at one time. So this is the spatial summation that we talked about. The other type is the temporal summation, is addition of stimuli occurring rapidly at a single synapse. A single synapse. So every time an action potential arrives at the synapse, another group of vesicles discharge acetylcholine, the neurotransmitter, into the synaptic lift. So every time more neurotransmitter, here acetylcholine, arrives at the postsynaptic membrane, more chemically regulated channels are open, the degree of depolarization will increase, so series of small steps can bring the initial segment to threshold and cause development of action potential. We can find in the illustration in the figure C, these are multiple stimuli at a single synapse. This is the temporal summation. Okay, then summation of excitatory postsynaptic potentials and inhibitory ones. Inhibitory postsynaptic potential is like excitatory postsynaptic potential stimuli. They spatially and temporally can be summed, but inhibitory postsynaptic potential is activation of a different type of ion channel. So the antagonism of inhibitory postsynaptic potentials and excitatory postsynaptic potential is important for cellular information. From this figure, we can realize the presence of excitatory neurons and inhibitory ones, presynaptic. So excitatory neuron 1 and 2 and inhibitory neuron 1. Under the figure A, we can see a stimuli from excitatory neuron 1. Then after some time there is another stimuli. There is no addition or no summation here. Under the figure B, we can see excitation for the neuron number 1 followed by a second one from the neuron number 1, excitatory neuron 1. This is the temporal summation. While under C, we can find the effect of the spatial summation when there is excitation from neuron excitatory 1 and the excitatory 2. While under D, we can find the integration, or here the cancellation, when there is summation between excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Okay, this figure also illustrates the same meaning when we have excitatory postsynaptic potentials, and if there is an addition of an inhibitory one on the left with the red color, how it cancels the development of action potential at the initial segment. Another definition here, facilitation. So spatial or temporal summation of excitatory postsynaptic potentials will not necessarily depolarize the initial segment to threshold. That's correct. But every step closer to threshold makes it easier for the next stimulus to trigger an action potential. So a neuron that has been brought closer to threshold is facilitated. The larger the degree of facilitation, the smaller of the additional stimuli needed to trigger an action potential. In a highly facilitated neuron, even a small depolarization stimuli will produce an action potential. So from this figure, we could see that neuron C and E and the relation between them. When neuron E facilitates neuron C, we can see that the combination between this stimuli can generate an action potential. An example for facilitation, for example here, nicotine. Nicotine stimulates postsynaptic acetylcholine receptors, producing prolonged excitatory postsynaptic potentials that facilitate CNS neurons. Another example, active ingredients of coffee, colas, cocoa, and tea, which are caffeine, theobromine, and theophylline, cause facilitation also. They lower the threshold at the initial segment, so a smaller than usual depolarization will cause an action potential. They also increase the amount of acetylcholine, the neurotransmitter, released, so it will cause more channels to be open. So these are nice examples for facilitation. Chloride channels and excitatory postsynaptic potentials and inhibitory postsynaptic potential summation. The chemicals that inhibit excitatory postsynaptic potential formation open chemically regulated chloride channels rather than potassium channels. Why? Because the equilibrium potential for chloride channels, or chloride ions, is minus 70 millivolts. With the chloride channels open, any shift in the transmembrane potential to, for example, minus 65, which is above minus 70, chloride ions start rushing into the cell. So it will acquire a much larger than normal stimulus to depolarize the membrane to threshold when chloride channels are open. Here we can see the excitatory postsynaptic potentials where sodium ion flow inwards is responsible for the generation of excitatory postsynaptic potentials. And here we can see that chloride channels or chloride ions flow inward also is usually responsible for the generation of inhibitory postsynaptic potentials. Another subject regarding presynaptic inhibition and facilitation. Here, the axo-axonal synapse that occur on a synaptic knob can alter or modify the rate of neurotransmitter released from the presynaptic membrane. For example, the presynaptic facilitation activity at the axo-axonal synapse increase amount of the neurotransmitter released when the action potential arrives at the synaptic knob. For example, serotonin cause voltage-regulated calcium channels to remain open. So arrival of action potential will cause prolonged opening of calcium channels, more calcium will enter, more neurotransmitter will be released, and the effect will increase on the postsynaptic membrane. We can find the illustration here, At the right side, we can see the presynaptic facilitation, when action potential arrive at the presynaptic neuron, and also excretion of serotonin, the neurotransmitter at the axonal synapse, will cause prolonged opening of calcium channels. Calcium will enter, the action potential arrives from the presynaptic neuron, more calcium will enter, more neurotransmitter will be released, and there will be increased effect on the postsynaptic membrane. While in the presynaptic inhibition, GABA as a neurotransmitter inhibit the opening of voltage regulated calcium channels in the synaptic nerve, causing decreased amount of neurotransmitter released by the presynaptic membrane. Then what happens here, action potential will arrive, fewer calcium channels will open, less calcium will enter, less neurotransmitter will be released by the presynaptic membrane, and the effect will decrease on the postsynaptic membrane. We can see the illustration on the left. When action potential arrived at the synapse, the action potential arrived, less calcium entered because the presence of GABA at the axonal synapse, then less neurotransmitter is released, and there will be reduced effect on the postsynaptic membrane. Thank you for your listening. If you have any question, please send them through the academic profile. Also follow my lectures on the YouTube channel and Google Classroom. Bye-bye.